No, I'll be alone in New York.
Oh.
I know.
Sounds like there must be a song or a movie, Alone in New York.
Alone in New York, working on mitochondria.
It's exciting.
It's gonna be great.
Right, I think it calmed down now. Everyone is here. Welcome on this Monday morning to Neuroccino. Feels like I haven't seen you in ages, but it's just been a week where Victor got his PhD. Woo! Congratulations. Today we have Ambra present to us. Ambra is a postdoc here at the Max Planck Institute and a very recent Marie Curie Fellow. And I don't actually know the paper she's going to present, so I'm just going to give the floor to you. Ambra, take it away.
Yes, thank you. Can I share my screen? I guess so. Yes, I can. So let's see. You should be able to see my screen in a moment. Yeah, I hope so.
Yes, and, do you mind zooming in a little bit so we
yeah.
have full screen?
Better?
Perfect. Thank you.
Yeah. So today, first of all, thank you for having me here at the Neuroccino. It's my first time, but I watched a few that you have published online. And so I thought that this preprint that I saw published a few weeks ago could be of interest to this group because it presents a new way to use hyperalignment to get category selective topographic maps in the human brain. And basically it leverages the technique of hyperalignment, as I said, based on connectivity measures to derive these category selective maps without having to use functional localizers pretty much. So first of all, let me guide you through the introduction of the preprint. And I'm sorry, of course, the layout is not very reader friendly because it's still a preprint, but I'll do my best. So the starting point is that category selective topographies are, well, of course, we know that every brain contains these category selective responses. For example, think of the ventral visual stream. We have face selective areas, body selective areas, place or selective areas, and so forth. And even though these are similar across individuals, of course, they're also highly idiosyncratic in terms of exact location. And so what we've been doing so far has been to have functional localizers for each individual participant that take part in our studies aside our main experiments of interest. And this is what I've been doing, for example, during my PhD and my postdoc. So my background is in functional MRI. And yeah, if you have a research question that involves a functionally defined area, for example, I worked with the LOC, the lateral occipital complex, which is sensitive to object features. What you have to do is that on top of your, as I said, on top of your main experiment, you have to spend time and energies on running functional localizers, which tend to take, well, at least 10 to 15 minutes on top of your other tasks. So here, the idea is instead to see whether we can avoid using functional localizers and instead use hyperalignment by bringing localizer data from a normative sample to the brain of our specific participants. And I thought that before diving into the details of this of the technique they used here specifically in this new preprint, I thought it would also be nice to briefly mention in general what is hyperalignment. And now I hope you see the eLife paper. So this is a recent review paper, which I highly recommend if you're interested in the method. It's been written by James Haxby and colleagues. Haxby was the first one who, well, his lab developed the technique pretty much, and the first publication goes back to 2011. And here they review the technique and they also compare response hyperalignment to connectivity hyperalignment. So the first one relies on, well, on brain activity to specific stimuli. Instead, the second one relies on the connectome. So you can, on the one hand, hyperalign brains based on brain responses, but you can also do it based on connectivity measures. And so briefly, here, the general idea of hyperalignment, which, as I said, applies to both measures, activity or connectivity. The idea is that information is shared across brains, and so what we care is to basically align brains, not in terms of anatomy, which is the standard pipeline in fMRI, but we want to align them based on a feature space, based on information, pretty much. And so basically we bring brains into a common high-dimensional information space. So that's the general, you know, two words definition of hyperalignment. And in practice, how is this achieved? Let me show you a figure which hopefully is more intuitive. So we start on the left from cortical maps in each participant. So participant 1, S1, participant 2, S2. What happens is that we have these cortical maps, which we can basically transform into matrices where each row is a time point, for example, T1, and each column is a voxel. So basically, if you look at a column, you see how activity in a specific voxel varies over time, or you can also see what kind of pattern of activity you have across the brain at a specific time point. What happens is that we want basically to bring these individual data matrices, which of course vary from one participant to the next. We want to bring them into a shared common space, where the coordinates are not like X, Y, Z in space. We're not bringing them, let's say, from individual anatomical space to the MNI space. No, what we are doing here with hyperalignment is to bring them from their native anatomical space into a feature space where each different dimension or vector here, D1, D2, D3, and so forth, represent a, if you want, a direction in information space. For example, could be, I don't know, if we are watching a movie, which is what people generally use when they do hyperalignment, because you can basically sample from a very rich pool of stimuli. You have, for example, in one dimension faces, in another dimension bodies, in another dimension again you have natural scenes, and so forth. And so the idea is to see how much a specific voxel contributes to a specific feature or dimension once it is projected from anatomical space to this high dimensional feature space. And to be even a bit more specific, let's also look at the matrix transformation. That basically, this is a matrix multiplication that basically brings the data from the individual brain space. Here on the left, you have matrix B. And so I bet people are familiar with this type of matrix, because you have, again, voxels on one dimension and time points on the other dimension. And by multiplying this matrix with the so-called transformation matrix, we end up into this common model space, where now we have on the top dimension, instead of having voxels and so anatomical space, we have this high dimensional feature space. If you have any questions so far, I'm happy to take them, because I would like this idea to be clear, and then we can move on to the preprint. So if there are any comments or questions, I'm happy, and I'll try to clarify. but maybe I can walk you through one row of the matrix. So for example, let's take T1.1. So we have this vector of voxels, and we multiply it by, so we multiply this row in voxel space with this column of the transformation matrix, and by then summing, basically it's like it's a weighted sum. By summing the result of this multiplication, we end up with the first item in the final matrix. So just to summarize, multiplying these matrices transform the individual brain data matrix, which is B, into the model space coordinates M, which is the final matrix. And of course, elements in the brain data matrices are measures of local neural activity, if it's response hyperalignment, or they are connectivity measures if the initial in this initial matrix, if we are doing connectivity hyperalignment. And then elements in the transformation matrix here in the middle are these weights, which basically capture how much a particular voxel contributes at a specific time point to the to a specific dimension in the hyperspace. And then finally, data in this indeed in this final matrix M are weighted sums of measures from the cortical coordinates of the initial brain space matrix. Okay, so I know hyperalignment can be a little bit tricky to understand and also for me to describe, but I hope that we capture the basic idea of hyperalignment. We bring individual participants' brain data, which are in native space, into a common hyperspace, which describes features, information. And as I said, then I really want to go back to the preprint. What we can do is response hyperalignment or also connectivity hyperalignment, which is at the core of the preprint. So connectivity hyperalignment requires the initial matrix. It has to be a matrix of connectivity pattern vectors in which each column is a cortical location in a cortical region to be hyperaligned, and each row is a pattern of connectivity. And the crucial point, the last point that I want to take from this earlier review is that whereas response hyperalignment depends on, of course, on response pattern vectors that are time-locked to specific stimuli, instead connectivity hyperalignment depends on connectivity vectors that, they do not require a common stimulus paradigm. So you don't need people to have watched, for example, the same movie, because you don't need responses that are time-locked to the same stimuli. And so this basically allows connectivity hyperalignment to be more flexible because you can use it across data sets, across different experiments. And this is exactly what they leveraged in this new preprint. And the idea was now to use connectivity hyperalignment to derive, as I said at the beginning, category selective functional topographies without having functional localizer data for each individual participant. And how do I take this now? Maybe I can briefly read through the intro and then go into the methods. Okay. So in previous work, they have established response hyperalignment, as I said before. But again, response hyperalignment requires time-locked responses to the same stimuli. And instead here, they wanted to test indeed if connectivity hyperalignment can be used. Let me see now. Yeah, probably now I would like to show you exactly what they did. So unfortunately here they first discuss the results, but I would like to jump towards the end. Sorry about this. I'll go into the methods. So they had 4 different data sets because one of their aims was to also show that you can generalize from one data set to another, again, based on the idea that connectivity hyperalignment works even if you present people with different stimuli. And but in each data set, and it's 4 in total, in each one people watched a movie inside the scanner. And then for each participant here, they also had localizer data because, you know, this is the standard method, like the gold standard that you want to beat. So basically, they also ran a standard functional localizer for faces, objects, bodies, and so forth. And then they compared the results that they obtained from these functional localizers, the traditional method, to connectivity hyperalignment. And so we have the Budapest data set. People watched Grand Budapest Hotel. And they also watched Raiders of the Lost Ark. So people had fun in the scanner. In the second data set, again, the same movie, Raiders of the Lost Ark, with the same scanner and same parameters. So basically, the only thing that changes here is the movie that people watched. And then they had forest data set. You might understand now which movie they watched. They saw Forrest Gump. And it was in with a different scanner, of course, different acquisition parameters. And then in the fourth one, it was again Raiders of the Lost Ark, but they used a different scanner again. So basically they had 4 data sets, and using a combination of these data sets, they could test whether the technique generalizes to different movies and also to different scanners, and so also different acquisition procedures. And the teaser is that indeed it does generalize. So they did very standard pre-processing using fMRI prep, nothing fancy here really, except that of course you do not want to normalize the data to MNI space. So you stay in participants, individual space. And then what you do is that you use the movie data to estimate the connectivity-based hyperalignment for each participant. So let me just briefly go back to my matrix here because it helps me a lot. Where is it? So the idea is that using the movie data, you, well, first, now I'm getting confused myself, but what I think happens here is that as a starting point here in the individual brain space, the X values are connectivity measures. So for a given voxel, you measure its connectivity values with all other voxels in the brain of the particular subject. And then you have it for each voxel and for each time point. What you do is that you do this for each participant, and then using the hyperalignment technique, you bring all participants into this same high dimensional space. Yeah, that's pretty much it. So basically here in the initial matrix, the X values are not response time locked values. They are connectivity values, functional connectivity values. And then, I mean, they also did response hyperalignment because this is a more established technique, and they also wanted to compare the two directly. And I'm skipping this, but basically the algorithm they used for this connectivity hyperalignment is advanced, but it's a lot of details, so I will skip it. Once they did hyperalignment, then they computed the category selective functional maps for each participant. And first, they did it using each participant's own localizer data, which, as I said, is the standard pipeline. It's like the gold standard that now they want to, they want to beat using the hyperalignment technique. And then, indeed, on top of doing this, they also estimated contrast maps using connectivity hyperalignment. So basically, instead of computing functional maps for each participant using data from that participant, they used instead data from other participants, thanks to the fact that now they had aligned all data to the same high-dimensional feature space with connectivity-based hyperalignment. Okay, so in the first case, we have contrast maps, functional maps for each participant using their own functional localizer data. Instead, in the second case, they derived the same contrast maps for each participant, but now using data from other participants. Let me see now if I have to add other details in terms of method, but I don't think so. No, basically, in summary, each participant's category selective maps were estimated based on the participant's own localizer data, and then all other participants' data projected into that participant's cortical space using hyperalignment. Oh, and I didn't mention, they also used a second method to compare, so basically anatomical surface alignment as a different way to bring people to the same space. Basically, instead of using hyperalignment, they also used, in a different control analysis, they used anatomical surface alignment. And you might expect which one of the two wins, given that functional maps vary across participants. If you align them based on anatomical landmarks, well, you have a lot of variability, and the maps tend to, blur and cancel each other out. So this control analysis, well, I'll tell you already the results, gave much worse outputs. This is it in terms of methods. Now I want to show you the results. Let me go back to the I think that is the first one. Yeah, so this is the first result image. So first, what we see in panel A is the functional topography here for one representative participant. And they zoom in into the ventral occipitotemporal cortex, because again, here they used the ventral visual stream as the target stream. We know that, FFA, LOC, PPA are well-known, well-established. So they thought, okay, let's test this method on a well-known cortical hierarchy. And here they show maps for FFA. So they contrasted faces versus all other stimulus categories, bodies, houses, and scenes. And this on the top left, you see the results using the own localizer data for this particular participant. So again, this is like the gold standard if you want. This is what I've done so far, basically. For example, and then they compare this one with the cortical topographies that they get using connectivity hyperalignment, CHA, and also anatomical hyperalignment as a further control. So of course, as I said before, if you align based on anatomy, you get blurry images. I mean, the broad topography, I would say, is pretty similar, which makes sense. But well, to me, this is also the output of anatomical alignment is also a, how do I say, like a way to demonstrate that what I've done so far makes sense. I've spent time, running functional localizers for each individual participant all the time. And this makes sense because if I didn't do so, I would come up with very blurry maps. But now, finally, the interesting bit here is what we find with connectivity hyperalignment. So remember here, what happens is that instead of using the localizer data from that participant. We use the localizer data from all the other participants, and we bring it back to the brain of this particular subject using the transformation matrix that comes out of the connectivity hyperalignment. And I think it's also fascinating because it demonstrates that the connectivity measures help us retrieve functional maps in a specific subject brain. So that's one reason why I was fascinated by this method. On top of the fact that it also demonstrates that I don't need function. Sorry, I keep saying this, but I think it's super great that it demonstrates that you don't need a face functional localizer, a body functional localizer, and so on and so forth. You just show people one movie, which of course has to capture a broad range of features, but then with one single 15 minutes movie watching experience, which is also, by the way, more pleasant for the participant, you can then retrieve cortical maps, category selective cortical maps, using this technique. So I liked it. In panel B, we have a scatter plot. We have, well, if you can see in colors, we have pink, yellow, cyan, and purple, and these are the 4 different data sets. So Budapest, forest, and so forth. Just to show basically that it replicates across data sets. And what is represented here in panel B, as I said, is a scatter plot where each dot is one individual participant. And on the x-axis, we have the correlation between the cortical topographies derived from the localizer data. And this is correlated with the cortical topographies derived from the anatomical alignment. Instead, on the y-axis, we have again a correlation between, again, the cortical topographies derived from the localizer data and now the hyper-aligned topographies. In other words, on the x-axis, you have a measure that captures how well the anatomical aligned maps worked, and on the y-axis you have how well the hyper-aligned topographies were. And as you can see, well, if the two worked equally well, you would have dots on the symmetric diagonal. Instead, we have that basically all the dots fall above this line, which means that the correlation between the localizer data and the hyperaligned maps were higher. I'm not sure if I described this scatter plot clearly, but yeah, feel free to ask me questions. But it's just a way to visualize the fact that basically the cortical maps using connectivity hyperalignment are better and more similar to the gold standard rather than the anatomically aligned maps.
I'm gonna stop you there because we're already at 10 o'clock.
Oh, okay, sorry.
No time for questions.
No time for questions. Well, this is the final plot that I wanted to show you because I think it captures the essence of the paper. Basically, yeah, I said it all. I don't know if you have any remarks.
There was questions in the chat already.
Okay.
People want to unmute. We have a little bit of time to go through them.
Okay. Maybe I can also stop sharing my screen so I can see things.
I can do that for you. Hang on. Stop sharing. There you go.
Okay, great.
Right, the floor is open. Questions?
I see. Shall I read in the chat or as you prefer?
I think it might be nice if people just unmute and ask their question. Michel, take it away.
I have a question. Thank you for this great presentation. It is really interesting. My understanding is like because you reduce the pattern or functional activation or connectivity to vectors, so like the statistical assumption that is behind it is we all have the same vectors and we can project ourselves with different intensity along that. And this is great, that cancel out a lot of noise and we get like those very sharp reconstruction. But I was wondering whether, not everybody functions the same, and you have categorical differences, even within healthy controls, but also in pathological states. So that might actually cancel out those differences. And one thing that I didn't see in your presentation, but might be in the material, is like when you use those vectors, you can actually obtain a number, which is a percentage of explained variance that you have per subject. Do they in any way provide that number for the participants they decode, or like,
Yeah, so with the idea that you don't know what is the ceiling, like the noise ceiling, is that what you mean, that you don't know how much variance is actually explained by the method? Or...
Yeah, let's take if you were taking the brain of an alien, for example, put it in the MRI scanner, watching that video, and use those vectors to explain, you will certainly have values that come out as significant, but they will not be representative of the functioning of that brain.
I see. Yeah. So one thing they did, of course, I mean, by comparing this method to the own localizer data, at least, I would say that is the gold standard. That's the measure we know captures the functional profile of that specific participant. So I would say, and then they also use the measure Cronbach's alpha to measure the reliability of the own localizer data, and that provides a ceiling, a noise ceiling that is like, okay, this is the maximum explained variance that we can capture using the own localizer data. And so this is what the other method, the fancy one, the connectivity-based method, that's what that one should also, that's where the other method should also reach this level at least, to so that we can basically conclude that this method is also robust and gives us meaningful results.
That is cool.
But yeah, so that's what they did.
That is cool.
Yeah, because of course you have multiple runs of the functional localizer and there's some noise associated with it. But then if you basically get a measure of internal consistency, of reliability, which is this Cronbach's alpha, then they compared the other methods. Yeah, using these metrics. Yeah.
Thank you.
You're welcome.
The day you scan the alien, let us know.
But yeah, if I can have one final remark, I think this
final remark.
Cool, because functional localizers are very boring. Static images all the time for 15 minutes on top of your main experiment. Instead, here you play a movie, and at the same time you get brain data for multiple categories at the same time, right? Which is also more efficient. People are more attentive, it's more entertaining, and that's also why they claim that it may be more user-friendly also for special populations, children, but also patients, people with a shorter attentional span. And then you need a normative sample with the functional localizer data, and then you back project this functional data into the specific subject space using hyperalignment. So that's why I got excited by this method. And I thought to bring it here because it also shows that connectivity-based measures are way more flexible because they abstract from the specifics of the stimuli, right? Because you don't need, as I said, time-locked activity to a specific movie. You can cross-generalize to different movies because the connectivity measure is more, it doesn't care about time-locked activity to a specific stimulus. So these are basically the two main reasons why I loved this preprint, and now I hope that it gets published and we can see if the method becomes if it takes I hope so.
Thank you so much for presenting it. I think there's definitely a benefit for special populations, as you said, with patients and children not having to spend that much time in the scanner. That's a plus for sure. Thank you so much. That's all the time we have. Have an exciting week in science, and we shall see you next week again for the next Neuroccino. The slot is still open, just letting you know. Let me know if you want to present. Thank you. Have a good week. Bye.
Bye-bye.